Welcome back to the OIS Retina Podcast. This week, Dr. Rahhal is joined by his friend and colleague, Dr. Steven Schwartz, who has an affinity for ophthalmic innovation and working with startups. Let's listen in.
Welcome back, everyone, to the OIS Retina Podcast. Thank you for joining us again. This is Firas Rahhal, partner at Retina Vitreous Associates Medical Group in LA and partner at Excite Ventures. And I'm super delighted today to have as my guest my very good friend and my neighbor here in LA, Dr. Steve Schwartz. All of you in Retina know who Steve Schwartz is. I've known Steve for, I guess, about 20 years, maybe more now. Steve maybe you'll remember better than me, but in that neighborhood, maybe more than that. Steve, as most of you will know, is the director of the retina division at UCLA Stein, has been there for many years. While he's there, he's of course training residents. He runs a very large clinical, surgical and medical retina practice, does a lot of surgery, lots of clinical research, lots of scientific research, lots of management. He's involved on the corporate side, as we well know, and he's also involved in a lot of innovations and venture and investment, which is how we've come to have him on. We want to hear about some of those things. We want to hear about what he's doing. And I'm involved in some of those activities too. So Steve and I talk frequently and share ideas, and he's always very generous with his time and with his ideas. Steve, welcome to coming on to the program, and thanks for taking the time to spend 30 to 60 minutes with us.
Hey, thanks for having me, Firas, and congratulations on this fantastic series. It's really a pleasure and an honor. You've had some great guests, and it's really you're scraping the bottom of the barrel now, buddy.
No, you're still in the first 6 months. You're like on the top of the barrel, but I guess I'll have to think what I'm going to say to the guy 3 years from now, if we're still doing this. When did you come to me? So tell us, I went through some of the many activities that you do at a high level, and I'm close here to you geographically, so I know about them, but you tell us in your words, like, what brought you here? How do you spend a week? What are the things you're doing that take up the time? And what do you enjoy the most, if you can share?
Sure. Well, I guess my story is a road well traveled. A lot of people have been down this road. I started in college, really interested in innovation and solving problems. And I was at Cal, Berkeley, UC Berkeley, and started working for some professor. I heard a lecture, and he was, his name was Ed Wilson. He was looking at innovative ways to try and figure out how closely related man is to different animals. And we used mitochondrial DNA and with a fixed mutation rate, and sort of did the math and backfilled it. And he came up with what was then a really revolutionary, at that time, theory about being 1% different than chimpanzees and gorillas and 2% different than monkeys. And everybody who's old enough remembers this. And at the time, there were these huge debates going on around the country. The religious right was all about intelligent design and creationism. And there were these creation versus evolution debates. And I didn't get into any of that. But it was really fun. And what I learned from that is, and there were really smart people. There was another guy there called Jim Dietz. And I didn't even know what I was going to study at that time. I was playing sports and chasing girls and being a usual, college experience. And I got exposed.
Now, I guess, I don't know. Is this, no, you've given all that up now.
I'm an old man now, my friend. But the takeaway is I learned some really interesting things from him. And it's really the 3 I's, innovation, impact, and integrity. And that sort of mantra has driven my career and where I've gone. And I went from ending up studying archaeology at Cal, origin of the species type of stuff, to not really wanting to go to medical school. I applied to one medical school. I was lucky enough through my father to know a guy called Howard House, who was the innovator of the cochlear implant here in Los Angeles. And Howard was an absolute Renaissance man of the highest order. He was a great surgeon, a great doctor, had started the House Ear Institute, innovated the cochlear implant, was a great lecturer, had a he was in his, 80s when I met him or got to know him. He had a gorgeous girlfriend who was a famous actress by the name of Nanette Fabray, who was about, I don't know, 20 years younger than he was. And he used to always tell great jokes, and he just enjoyed his life so much that he talked me into applying to medical school. So I applied to one medical school, and mostly because he made me laugh so much. He was the king of the old medical school jokes. He would pull his something out of his pocket and look at it and say, "Oh, my thermometer. Now I know where my pen is." And, and it was just like never ending. And so I ended up through some fluke getting into medical school, and
That's interesting. That took guts.
Yeah, and I went, and I got to USC, and they randomly assigned mentors at USC, and my faculty mentor was a guy called Steve Ryan.
Oh,
and, and so Steve met with me, and after about 10 minutes, he was like JFK. I mean, he was
larger than life,
so charismatic, and such a leader and a great guy. And he basically said, make good grades, get good test scores, write some papers. And by the way, I want to introduce you to this guy next door. His name's Ron Smith, and he's going to be your mentor. So he pawned me off in 2 seconds, and did some research with Ron. And at that time at USC at Doheny, the group was really interesting. There were some really great retina docs. It was a revolving door in those days for no good reason, or reasons that were above my pay grade. But in cornea, the fellow, and then the early faculty member was Peter McDonald, who is now the chair at Wilmer, and Peter Liggett was in retina there, and John Lean, who ran the silicone oil study, and then Zden Greger, and all these great people. And so it was a really, really fertile, exciting place. And I couldn't decide whether or not to go into orthopedics. I really liked sports and sports medicine. I'm sort of a broken jock, and I had a chance to work with Dr. Rosenfeld, who used to be the team doc for the Raiders in the Al Davis days, and it was really a fun time. And I was really leaning towards going into ortho, which was sort of flash your garage and pull out the drill and fix people. It was a really fun time.
Chisels, and for athletes, it seems to be a natural fit. Did you play ball in college?
A little bit. I was pretty bad, and I remember. Well, anyway, it's all the sports stories are. We got to play "Glory Days" by Springsteen in the background, and we can talk about it. But no, is the take-home answer. No, nothing of note. Stanford had John McEnroe in tennis, and Cal had nobody, and Stanford had Elway, and we had in football nobody, and I was bigger than the whole offensive line, and it just wasn't going to happen. So my dad said to me, go to the library. Yeah. And I did. And anyway, I was in medical school and was leaning towards ortho, but I did a couple of research projects with Ron and Steve, looking at all sorts of things. That was the time of the trauma model with Cleary and the laser model for CNV. And there was a lot of exciting stuff happening right downtown LA at the county hospital. And Doheny was growing at the time. And so I did a project with Ron, and then he said, he pawned me off to Bart Mondino over at UCLA, who was the cornea guy at the time. And I came over to Bart's office, and I was a USC undergrad, and Bart said, no. And I said, well, yes. And he said, no. And so I didn't know what to do, but I was just persistent by nature. So I went around and found this other guy who was a young glaucoma doc, and he was collaborating with Bart. And a week later, I was in Bart's research meeting, and Bart's like, how did you get here? I kicked you out. Ron had,
I thought I said no.
I thought I said no. It's like that Mike Myers thing from Austin Powers. How about no? And so we did this research project looking at Bausch and Lomb project. They had they pawned me off on everybody. I ended up working with the optometrist at UCLA, and he was a contact lens expert, and we discovered how to calculate how fast or how efficiently oxygen transits contact lenses. The formula was dK over L. And we figured out why you shouldn't sleep in your contact lenses, why people who slept in their contact lenses got corneal ulcers. And I remembered being at the county hospital as a medical student and watching one of the senior residents do a subconjunctival injection on a unit patient, putting some antibiotics under the conj because they just weren't getting their drops.
Yeah.
And the patient had been like asleep in a coma for months and woke up and screamed with a subconj injection. So
It's a miracle.
I took these collagen lenses that were disposable and dissolvable and impregnated them with vancomycin and amphotericin and other drugs, and it got huge play at the academy and huge play at the in the cornea world, and it was my first real taste of innovating. And then I was working on some other things. I had a tough, and then I was lucky enough I applied to both ophthalmology and ortho, matched at Jules Stein, super lucky. First person they'd ever taken from the little sister university across town. I was the.
Is that true? Nobody had ever gone over?
Well, Krieger was an undergraduate at USC, and Bob Christensen was an undergraduate at USC, and they were always. Krieger still has a Trojan football thing in his office, with letters from Pete Carroll and Reggie Bush and everything. But that's another story. So I crossed town and had a not great experience doing research with a faculty member in retina who's no longer at UCLA and had left shortly after I graduated my residency. And there was a lot of innovation in that project and potentially a lot of impact, but there was no integrity. And the paper ended up getting withdrawn from ARVO, and I ended up getting pretty upset about it. It was hard on me as a resident to have a role model who was closer to my age not behave well. And that experience, and I knew I was going to go into retina, and I was really fortunate because one of the great minds in retina was taking a sabbatical a couple of years at UCLA in the lab. His name's Alan Bird, and Alan was working in Dean Bach's laboratory. And Dean Bach's laboratory, as a graduate student, he worked on describing how photoreceptors shed their outer segments and the visual cycle and work into the RPE and come back around and rejuvenate the visual cycle. So it's a really interesting laboratory. And Alan, even at this time, which was a long time ago, firmly believed that there had to be a better way to treat neovascularization, whether it was from the choroid or from the retina. There had to be a better way to treat it than physically burning it with a laser or surgically removing it. He just thought that was insane, and that was his big mantra then. And so that was the time the interferon study was just starting. And anyway, I went to London to Moorfields Eye Hospital for my fellowship, and that was an incredibly fertile place. My co-fellows at Moorfields were Frank Holtz, who's a giant innovator in AMD, and Alex Harper, who's probably the finest medical retina doc on the planet. He's in Melbourne, and a number of other really, really great people, including Jack Wells and a few others. And then I really broke the mold by applying for their surgical fellowship. So I was, interviewing at Duke and at Royal Oak and at all the great East Coast fellowships at the time. And I interviewed for the surgical fellowship at Moorfields for Zdenek Gregor's spot. At that time you were attached to one mentor, and Zden had been on the faculty at Doheny, probably the best vitreoretinal surgeon I've ever seen in my life. And certainly right there with Stanley Chang and a bunch of the greats, and Buzz Krieger and Brooks McCune and Mike Tracy, and just the really, really great surgeons. And Zden was an extraordinarily thoughtful guy. And in those days they had residents or senior registrars could stay on the house indefinitely. You could stay in your senior registrar job for as long as you wanted until you got the job you wanted. So you could, these guys, there would be jobs that would come up as what they were called consultants jobs in Blackpool or somewhere else. They never applied for those. They waited for the London jobs. And so Zden's job came up, and these guys had been fellows for years and senior registrars for a long time. And I remember that interview like it was yesterday. It was in an old giant boardroom at Moorfields Eye Hospital. It was like Hogwarts. And there were about 6 people behind the table, and I'm down at the end of this long conference table. And they asked me all these questions, like ridiculous things, and I was trying to be self-effacing and humble and thoughtful and so on. And then I finished. And in those days, the tradition was, it was such a big interview that you would just take the whole day off of work. And
Right.
being an American, I had A, no idea about that tradition, and B, I was there to work. So I basically left the room and then stuck my head back in the door. I said, if you need me, I'll be back in clinic. And I left. And I think that's how I got the job, because I was
Oh,
sort of
really?
a workaholic. And
They're impressed with the work ethic.
yeah, exactly. And so, working hard is important, but the drama of me getting the job is the point of the story. And that is academics is great for a lot of reasons that all of you probably know. You have a chance to innovate, you have a chance to take care of patients, you have a chance to teach, you have a chance to move the field. But there's a lot of drama and emotion in academics over very little things that are not impactful. Who's going to be an author on a paper? What order are you going to be in? Who's got which room? Who's got this? Who's got that? It's a lot of politics and drama. And that really is not why I'm in academics. I'm in academics for the opportunity to innovate, to be impactful through those innovations and through teaching and of course delivering patient care and moving the field that way, and to try and be a beacon of integrity. Because as we get older, the young docs have to take care of us, and we need people that are going to come out. I'm extremely proud of our cohort of fellows that we've trained and residents. They're pretty good, and they typically get their first choice of jobs, whether it's residents coming out and getting great fellowships, or fellows coming out and getting great jobs. And so I'm really proud of our group. And so that's kind of how I got to UCLA. And then when I got to the faculty at UCLA, it was so busy when I got there and I had such a busy surgical fellowship. And I brought with me some things from Europe that hadn't really gotten to the States yet. For example, silicone oil was approved in Europe when I was there. So at Moorfields, I did hundreds of silicone oil cases. I got back to UCLA and they were still in the silicone oil trial. So I had a huge experience with that. PFL was not approved in the States. Stanley had it in New York.
Yeah, we had it in New York. We called it Stanley's juice. I still call for it that way now.
Right. So Stanley's good friends with Stan, and Moorfields made their own PFL. So we had like a little hose coming out of the ceiling. So I had a huge experience with PFL. And the other thing was we developed the wide angle viewing system with Stanley and Avi and with Volk. And so Dominic McHugh and I wrote the first papers on the wide angle viewing system out of Europe while Stanley was innovating that here. So when I got to UCLA, I had wide angle viewing, PFL, and silicone oil before anyone except maybe Stanley. And so I got really busy really fast, and took me a couple of years to come up for air. And that's how I got into it. And then I realized over time that, we could do a lot of innovating inside and outside the university, and big things happen at universities, a lot of resources and a lot of collaborations and stuff. And so one of my, I think, biggest contributions was during the submacular surgery trials. Neil Bressler was the chair of that trial, not a surgeon.
Not a surgeon,
Not a surgeon, but a great guy, good clinical trialist. Learned a lot from him and from Barbara Hawkins and the other people at Wilmer and the NEI in those days. But one of the things that I helped spearhead on the early days of the executive committee was bringing private practices into clinical trials. So when I called Dave Boyer, your partner, and said, look, Dave, I want you to be in these clinical trials. At first he was reticent, and now it's a giant piece of what you guys do. And you guys make, when I say you guys, not just Retina Vitreous here in LA, but all of the private practices around the country make enormous contributions to the field and move the field a lot faster through participation in clinical trials. And that really came to fruition when David Geyer and Sam Patel, Tony Adamus and I, and Mark Blumenkrantz, Joan Miller, that group of people realized that anti-VEGF was going to work, and that Genentech had this drug. And what happened was, independently, David went to Genentech, Sam went to Genentech, Tony went to Genentech, Joan Miller went to Genentech, Mark Blumenkrantz went to Genentech, and I went to Genentech. And we were trying to convince them to use what later became Avastin or something like it in the eye. And interestingly, the story around the anti-VEGF innovation comes from a guy who you know, but a lot of people don't, by the name of Richard Casey.
Mhm.
Richard Casey grew up in Compton, went to Harvard, started doing research as a medical student with a guy by the name of Judah Folkman, who clearly would have won the Nobel Prize had he lived. And he did the first anti-VEGF experiments in the eye in Judah's lab. And when he graduated his cornea fellowship, he passed his project on to Tony Adamus. And Tony, the rest is history. Tony took it and multiple touchdowns with iTech, with Ophthalmology, with obviously at Genentech Roche. Tony's been a huge innovator and champion of innovation and drug development. And, Richard and I are very close, and Tony and I are very close. And so that was what got me interested in. Basically, I got to hand it to David and really Samir, but David Geyer just said, look, we're not going to move Genentech fast enough. Let's do it ourselves. And he went and got the aptamer, which became Macugen, and, wrote the first papers and, we learned a lot of interesting lessons. I think had there not been so much pushback for intravitreal injections, and had David been willing to go with Q4 weekly injections, the Macugen data might have been very different, but still not as strong an agent as what Lucentis turned out to be, or now Eylea, and even more of them now. But the innovation there to get it going was phenomenal, and to get to work with Tony and David and Samir at iTech, and then we brought Emmett Cunningham in, and Emmett was awesome, and the rest is history. We got that drug approved and developed, and there was great basic science, and it was at that time that we brought all the private practices in. I was able to call my friends around the country and say, look, you should be in this trial. And to this day, that group has one of the best clinical ops groups in the country. Evelyn Harrison runs this amazing team. Some of her team have left and gone to Oyster Point and gone to other companies, but they're just fabulous. And they do a great job. Now, of course, they have Iveric, which is rewriting dry AMD. And it's just great to watch that all, come together. And so I guess for us, that's a long answer to your question of how did I get into this? Yeah. But through that experience at iTech and learning from those people, all of whom I've learned a ton from, I learned most importantly, I think, how to listen and how to integrate the lessons into my daily activities, and met a lot of amazing people along the way, and been able to participate in a lot of really exciting and interesting projects and ventures that I think have changed the field, which is my goal, to help patients, which is ultimately all of our goal.
I want to ask you about one other thing, and the gene therapy, we could spend the next 20 hours on it with all that you've done and all the things that are going on, but there's another project you're involved in right now. Before we finish, I want to give you a chance, and it's a project I find very interesting myself, the robotic surgery project that you're involved with, JP Hoopschman's involved with at UCLA. That's still in early stages. What can you tell us about that? Where is it? Where's ophthalmology going with robotic surgery?
So that's a great question. I think it's really important, the takeaway from the long stories I just told you is it's super important to surround yourself with great people. And if for people who are watching this and trying to figure out how to get started in this area, get yourself around great people and high integrity people if you really want to make an impact and learn how to innovate, learn how to fail fast and all that stuff. Well, the JP story, JP Hoopschman, for those of you who don't know Jean-Pierre Hoopschman, he's one of my partners at UCLA at the Stein Eye Institute. Many years ago, they still do this in France, by the way, they rate their surgeons. So you can look online and find out 1 through 25 who are the best rated vitreoretinal surgeons in France. It's ridiculous. It's ridiculous. So JP was rated 3rd, I think, Didier and one other guy and JP. And anyway, I didn't know any of this. JP came to me at UCLA and said, look, I want to emigrate. And he's a really special guy. And long story short, we were able to, over the course of the next couple of years, get him recruited to our department. When he got to our department, he was an international fellow for a couple of years until he could demonstrate the necessary steps and so on to get his California medical license and join our faculty for real. But So he was my fellow, but he knew more than I did. So talk about associating with great people. I've had a string of these fellows from Europe and Israel and Japan, and who are mid-career, and they're just unbelievable. And Gadi Howell's another one. He's on the faculty at Doheny now. He worked in my office for 5 years. And then Hamid Hosseini, who's now in our group, who's just a walking Google. He's unbelievable. So JP, we have a center at UCLA called the Center for Advanced Surgical and Interventional Technologies, CASIT, and it was run by the general surgeons. And they had a Da Vinci robot down in the robotics lab, and they asked us to play with it. And I brought the fellows down there and said, guys, we've got this robot to use at our disposal. Let's start doing some papers and figure out if we can use this to do things with, if there's any advantage. And we wrote, I think, 10 or 12 papers on using the robot. And JP was a fellow at the time when it started, and there's an Australian fellow and an Israeli fellow and JP, and it was this great international group of junior mid-career guys, and they started innovating, and it became clear to JP that it wasn't going to work. The Da Vinci was too big, the fulcrums were too big, and so on. But he really was interested in, we were really interested in things. DARPA and the Defense Department were interested in removing metallic foreign bodies from blast injuries in the wars in the Middle East. And the improvised explosive devices were devastating our troops. And, the way they were treating them was they put a little neosporin in the ruptured globe and a couple of iPads and some duct tape around their head and fly them to Germany. Yeah. So the results weren't great at the beginning. They've obviously been refined and gotten a lot better, but we were looking for a battlefield forward, a war forward robot. The Army has big bandwidth. So, we're looking for a surgeon here and a machine there with a flight surgeon setting it up and getting the metal and the eye repaired in the field, rather than, 24, 48 hours later, which you all know is rough. And so JP basically got with engineering and physics and computer science, some really amazing people. He's got the guy who fixed the Hubble Space Telescope remotely. He's got the guy who was chairman of mechanical engineering and mechatronics, and basically is the world's expert on micro machining things, builds things like the smallest chips, like ridiculous genius people.
Dr. Rosen?
Jacob Rosen is an Israeli engineer who built the battlefield exoskeletons for the battlefield.
My son took some classes with some of these guys. He got his mechanical masters at UCLA.
Yeah, Jacob's pretty impressive, but TC Tsao, TC, I don't think sees the undergraduates too much, maybe the master's students, but TC
student for the guys.
Yeah, that's pretty awesome. So TC is amazing, and so JP put this in computer science and mathematics, and so basically what he wanted to do retina work, and then we switched it to cataract, and by switching it to cataract, we now have a robot that's NEI funded. JP's got 2 R01s. It's NEI. It basically is automated cataract surgery. You could think of it like your Tesla. We talk about it as we are starting to consider taking some money into this entity. It's like a self-driving car. We're at stage 2, 3 right now. We already think that we know from our work that our robot can do cataract surgery with a surgeon there. It's doesn't break capsule, can't break capsule because it's got no fly zones. It has real-time OCT, so it doesn't leave any residual lens material, so you don't get posterior capsule opacities. Perfectly centers the IOL, and you don't need a surgeon. I mean, the femto does the first 3 steps of cataract surgery. The robot does the next, 9 steps, and that's it. And cataract surgery is like a forehand volley. It's the same every time for 99.9% of the cases. And, the great cataract surgeons in the country, when we innovated together with Mark, the femtosecond laser, Mark and Daniel wrote the patent for the femtosecond laser. Most people don't know that came out of Retina in Stanford. All the great cataract surgeons said, we don't need a femtosecond laser. That's ridiculous. We do a perfect capsulorhexis. I don't need a laser to take out the lens. And there's still a few guys who don't want to use a laser to take out the lens. But, what it does is it doesn't really affect the top 5 or 10% of surgeons, but it takes the bottom 50% of surgeons and makes them in the top 20% of surgeons in terms of complication rates. And let's face it, everybody's got complications and everybody's got a different, rate. And so to unify this and make it more uniform and safer, and so I think automated cataract surgery is on the horizon. This machine really, really does it. And some really smart people have tried to do it. But, some of the innovations that we've been careful to incorporate, not reinventing how cataract surgery occurs. So we're not replacing the femto. We're not replacing the phaco. We're not even using proprietary femto or phaco. But we are using a docking system, which is, obviously all this is IP protected. We've got a docking system. We've got intraocular, intraoperative OCT real time. We've got huge signal processing around this thing. And it's pretty clean and pretty quick. And it's amazing. You just watch the cataract come out and watch the lens go in. And
this is incredible. Is this, what stage is it in now in terms of the clinical development? It's in animal models that you're doing?
Yeah, it's well past that. I mean, it's ready the prototype is well underway for the first human trials.
This is awesome. And this is definitely coming, right? I mean, is there any doubt that this will
But you tell me, do you doubt that? I mean, think about the indications for robotic surgery. I mean, the Da Vinci robot and that company have done a great job. Intuitive have done a great job. And basically all they showed was non-inferiority for prostate and other stuff. And it's taken off, and partly because of marketing, and partly because the public feels more comfortable, and partly because the Da Vinci robot adds a lot of value to the surgeon. You don't have a tremor. You have great lighting. You have great visualization. You have huge advantages with robotic surgery and all of that stuff. And you can't put it where you don't want it to go. In our case, the eye is at least as unforgiving as any human tissue surgically. So you can't repair the capsule once you break it. So what we've done is we've created a situation where, and again, all kudos to JP and his team. I got them started and stood back. And that's, by the way, a good way to innovate, and maybe another topic. They've built no-fly zones. So the instruments literally can't break the capsule. You can try to, and you can't break the capsule. And Jacob Rosen's got this surgical cockpit. You can sit in this cockpit. It's three-dimensional. It's like you're inside of a golf ball. And you can do the surgery if you want from inside and control the whole thing. You can peel membranes with it. It's incredible with haptic feedback, and you can train in it and so on. And
is that next moving into the posterior segment to try to do intraretinal work, subretinal work? What's next?
Well, there's some really good groups in Europe that are doing, subretinal injections of stem cells and gene therapy and things like that with a precise robot. The precise robot, and they're a very good group, and really, really, really excellent. And but we're not. I think cataract is where it's at, and right now it's the killer app for this robot. And certainly studying some posterior segment thing, you can do things with this robot that you can't do reliably. You maybe can because you're a great surgeon, but most surgeons can't do reliably, like cannulate small retinal vessels, or
that sounds pretty challenging.
or maybe do intraneurosensory transplants. So if you wanted to put a certain cell type into a layer of the neurosensory retina, maybe not underneath, but that type of thing, it's just sort of beyond human capability. And we've got all this magnificent data, and so it's really exciting. We're starting to talk to some pretty big collaborators here financially, and it's still a number of years away. I don't think people that are listening to this need to get worried about it, but it's coming. I mean, don't you think robotic surgery is coming? There's no stopping it. It's just like AI.
Ophthalmology, and we've run out of time, but I was thinking that the one thing we've never had in ophthalmology that the other surgeons mostly have is we don't really have tactile feedback, right? We have, at least in retina, strictly visual feedback, which is amazing. We have great optical systems to view the internal limiting membrane and so forth. But wouldn't it be great if a robot could amplify the tactile feedback that you could actually feel touching the internal limiting membrane in some way?
Come on over to the lab for us. Come on over to Jacob's lab, and JP will sit you down in the, actually, we've got a robotics center now at the Eye Institute. And JP you can come over and sit in the cockpit.
I want to.
And you can feel the membrane coming off as you peel it.
Yeah, that's amazing.
It's amazing. Well, what the truth is, what you do is, what they've done is ingenious. They've calculated the necessary force to remove the membrane. and they've calculated the necessary additional force to put a hole in the retina.
Huh.
And so they integrate those calculations into the tactile feedback, and you can learn from the feedback about when it's going to rip and when it's not. And it's amazing.
Yeah. No, it's coming, and I look forward to it. I don't think we should feel threatened by it. We should be excited to do this.
Well, I can tell you, we're not going anywhere. Surgeons aren't going anywhere soon. And I tell the residents and fellows this all the time. And, we have a big AI project going on, and people are always worried, is my job am I going to get replaced by a computer or a robot? And the answer is no. And the reason we're not going to get it replaced is because look at your smile. Patients need to feel your warmth and humanity and skill set, and algorithms aren't going to be able to pick up the things you pick up, all the time. So it's not a translator that tells you whether it's a stop sign in a different language, but cataract surgery is pretty binary in terms of what the steps are and the things that need to happen. And so I do foresee a time where, not too distant future, where people are not going to be doing too much cataract surgery, maybe in the next 10 years, 5 to 10 years. One of our great new cataract surgeons at UCLA is a guy by the name of Sean Lin. And Sean has an MBA from Stanford. And he told me the other day, he said, I knew when I took this job that I wasn't going to finish my career doing surgery because nobody's going to be doing it.
Interesting.
Yeah.
Listen, that's amazing. And this is a great place for us to stop, because obviously we could go on about the robotics and the stem cell and the gene therapies and the other things you're involved with. But I'm going to end there by saying Steve Schwartz, innovator, educator, academician, corporate guy, overall fun guy. And I really appreciate you coming on. You and I have known each other for many years. Again, we have a good time together. I look forward to hearing more from you personally when we meet up, and maybe even have you back on this to follow on the robotic stuff later.
Yeah, I think the key thing you said is having fun. This is hard. What we do is hard, and it takes a lot of work and a lot of attention to detail. And I've been super fortunate, and a lot of luck involved. But you know that you gravitate toward the right people, high integrity people who are the ones who cross the finish line and who have patients first, and making money for your investors second.
Right.
It's a privilege, though, and for us, it's such a great time. It's a great excuse to get to see you during these weird times, dude. This is just too much. So thank you.
Thanks, Steve. It's really been a pleasure, and I'm sure our audience is going to enjoy hearing some of the anecdotes and the science. And I'll meet up with you again soon, the two of us over a drink.
Look forward to it. Thanks, guys. Bye.
We hope you enjoyed this episode. Remember to subscribe to our podcast and leave us a review to help us attract more listeners. And stay tuned for the OIS 2020 Year in Review virtual program coming up in December. For more info, visit OIS.net. Until next time, keep innovating.